The human gut is far more than a digestive organ. It functions as a central hub that influences multiple systems across the body, connecting intestinal health to cardiovascular and renal outcomes in ways that science is only beginning to fully understand. This gut-heart-kidney axis highlights the intricate interplay between microbial composition, metabolic signaling, and systemic inflammation. Certain bacterial species, such as Blashia hydrogenotrophica, have emerged in research as potentially linked to higher cardiovascular and renal risk through their influence on inflammation, metabolite production, and gut barrier function. Understanding how dietary choices can modulate these microbial populations opens exciting possibilities for whole-body health, offering tangible strategies to reduce disease risk while simultaneously enhancing metabolic resilience. Blashia hydrogenotrophica and related species can influence host physiology through several mechanisms. Some studies suggest that an overabundance of these bacteria correlates with higher levels of trimethylamine, TMA, production from dietary choline and carnitine, which the liver converts to trimethylamine N-oxide, TMAO. Elevated TMAO has been linked to atherosclerosis, endothelial dysfunction, and chronic kidney disease progression. Furthermore, microbial shifts favoring such species may reduce the relative abundance of beneficial SCFA-producing bacteria, diminishing butyrate and propionate availability. These SCFAs are crucial for maintaining gut barrier integrity, reducing systemic inflammation, and modulating insulin sensitivity. In essence, a gut environment dominated by potentially harmful taxa can create a domino effect: increased inflammatory metabolites, reduced protective SCFAs, heightened oxidative stress, and subsequent impacts on the heart and kidneys. The promising news is that dietary interventions can meaningfully influence the composition and activity of gut bacteria. Foods rich in fermentable fibers, polyphenol, and prebiotics support the growth of beneficial microbes that compete with Blashia hydrogenotrophica and similar taxa. Whole grains, legumes, nuts, and a variety of vegetables provide substrates for SCFA-producing bacteria like Faecalibacterium prosnitzii, Roseburia, and Bifidobacterium species, helping to restore balance in the gut ecosystem. By creating a competitive microbial environment that favors protective species, these foods can reduce the relative abundance of taxa associated with higher cardiovascular and renal risk. The metabolites produced by beneficial microbes, particularly butyrate and propionate, strengthen the intestinal lining, reduce endotoxin leakage into the bloodstream, and downregulate inflammatory signaling pathways that contribute to vascular and kidney injury. Polyphenol-rich foods such as berries, green tea, cocoa, and certain nuts also exert selective pressure on the microbiome. Polyphenol are metabolized by gut bacteria into bioactive compounds with anti-inflammatory and antioxidant properties, and they can inhibit the growth of less desirable bacteria while supporting SCFA producers. For example, pistachios and walnuts supply both fiber and polyphenol that encourage microbial diversity, creating a microbiome that favors cardiovascular and renal health. The synergistic effect of polyphenol and fiber is particularly compelling. Polyphenol can modulate microbial metabolism, while fiber provides the fuel for SCFA production, together reducing inflammation and promoting metabolic homeostasis. Fermented foods, such as yogurt, kefir, kimchi, and sauerkraut, introduce live probiotic species that can reshape gut ecology by outcompeting potentially harmful bacteria. Regular consumption of these foods enhances microbial diversity, increases SCFA output, and may help decrease pro-inflammatory metabolites like TMAO precursors. When combined with a fiber-rich diet, fermented foods create an optimal environment for a microbiome that supports systemic health, protecting the heart and kidneys through both direct and indirect pathways. Lifestyle factors further amplify these benefits. Consistency in diet, attention to portion sizes, and timing of meals can reinforce favorable microbial shifts. For example, distributing fiber intake across meals ensures steady SCFA production and minimizes postprandial glycemic and inflammatory stress. Pairing prebiotic foods with polyphenol-rich fruits or nuts can amplify microbial fermentation, while limiting processed meats, refined sugars, and ultra-processed foods help suppress the growth of bacteria linked to TMAO production and other inflammatory metabolites. Over time, these dietary patterns cultivate a resilient, balanced gut ecosystem that translates into measurable reductions in cardiovascular and renal risk markers, including improved lipid profiles, lower systemic inflammation, and enhanced kidney function indicators. What is particularly exciting about the gut-heart-kidney axis is that it illustrates a holistic approach to health. By focusing on the microbiome, we can simultaneously support multiple organ systems. Strengthening gut barrier integrity reduces endotoxin-driven inflammation, which lowers cardiovascular stress and preserves renal function. Enhancing SCFA production improves metabolic efficiency, regulates blood pressure, and stabilizes glucose and lipid metabolism. Reducing the abundance of potentially harmful bacteria like Blashia hydrogenotrophica not only limits pro-inflammatory metabolite production but also frees ecological space for beneficial microbes to thrive, creating a positive feedback loop that reinforces health system-wide. In practical terms, cultivating a microbiome that supports heart and kidney health is both accessible and sustainable. A daily diet that emphasizes a wide variety of colorful vegetables, whole grains, legumes, nuts, seeds, polyphenol-rich fruits, and fermented foods can shift microbial populations toward protective taxa, decrease inflammatory metabolite load, and promote SCFA production. Even modest, consistent changes, such as replacing refined snacks with nuts, adding legumes to meals, or incorporating fermented vegetables, can have measurable impacts over time. These dietary strategies do more than feed the gut. They help orchestrate a multi-system health advantage, reducing cardiovascular stress, protecting kidney function, and supporting long-term metabolic stability. Ultimately, the gut-heart-kidney axis demonstrates the remarkable influence of the microbiome on whole-body health. By shaping microbial composition through thoughtful food choices, it is possible to reduce the prevalence of bacteria like Blashia hydrogenotrophica that may elevate cardiovascular and renal risk while simultaneously fostering SCFA production, lowering inflammation, and improving metabolic signaling. This integrated approach positions diet not just as fuel but as an active tool for cultivating resilience, longevity, and systemic wellness, offering a concrete, scientifically supported pathway to optimize heart, kidney, and gut health in tandem.